Good morning, everyone. Welcome to the fourth subconference of the Young Academy of Sciences Summit. I'm Sabrina Chan, and it's my great pleasure to be your MC today. The theme of today's conference is From Cancer Discovery Science to Therapeutic Innovation, and it is organized by the Hong Kong Young Academy of Sciences and co-organized by the Greater Bay Area Homeland Investments Limited and the Greater Bay Area Homeland Youth Community Foundation, with funding support from the Innovation and Technology Commission of the HKSARG. The organizer and co-organizers would like to thank the following parties for their support, without which the event would not be possible. They are the Innovation and Technology Commission and local academic partners and supporting organizations, including City University of Hong Kong, Hong Kong Metropolitan University, Institute of Professional Education and Knowledge, Nanyang University, the Chinese University of Hong Kong, the Hong Kong Polytechnic University, the Hong Kong University of Science and Technology, the University of Hong Kong, Hong Kong Applied Science and Technology Research Institute Company Limited, Hong Kong Cyberport Management Company Limited, Hong Kong Science and Technology Parks Corporation, Hong Kong Young Industrialists Council, and the Hong Kong Academy of Gift Education. The event aims to further promote scientific exchange among young scientists from different regions, creating new opportunities for collaboration and showcase Hong Kong's advantages as an international science and technology hub. Now, I would like to invite Professor Stephanie Ma, Vice President of the Hong Kong Young Academy of Science, to deliver the welcome speech. Professor Ma, please.
There you go. Okay, good afternoon. It's too early in the morning. Good morning, Professor Alice Wong, Professor Jun Yu from the Hong Kong Academy of Sciences, Professor Lai Wen, Professor Wei Lang Tam, our international speakers, distinguished guests, colleagues, students, ladies and gentlemen. On behalf of the Hong Kong Academy of Sciences, it is my pleasure to welcome you all to the fourth subconference of the Young Academy of Sciences Summit that's themed From Cancer Discovery Science to Therapeutic Innovation. We're delighted to see so many of you all here today, all united by a common goal, which is to advance the fight against cancer. This subconference is part of a broader initiative that aims to foster collaboration and innovation in the field of science and technology. The Young Academy of Sciences Summit is organized in partnership with the Greater Bay Area Homeland Investments Limited, the Greater Bay Area Homeland Youth Community Foundation, which seeks to empower young scientists and provide a platform for sharing knowledge and ideas. I would also like to take this moment to express our sincere thanks to the Innovation and Technology Commission of the Hong Kong SAR government. Their generous support has made this conference possible, and we're thankful for their commitment to fostering innovation and technology in Hong Kong. I'm also very honored to welcome Professor Alice Wong, the Associate Vice President of Research here at Hong Kong U, and also the Interim Director and Chair Professor of the School of Biological Sciences, Faculty of Science here, who has accepted our invitation to deliver the opening speech today. So Professor Wong herself is also a cancer biologist with a focus on signal transduction and in particular cell adhesion molecules. So her presence and insights are greatly valued. Today's theme is especially significant as it addresses one of the most pressing challenges in healthcare. We're very fortunate to have a remarkable lineup of speakers who will share with us on a wide range of topics on cancer, from prevention, early detection, therapeutics, and also technologies to approach the topic. We're also immensely grateful to our invited speakers, both local and international, for taking the time to share their experience with us today. I encourage everyone to engage actively, ask questions, and also take most of the networking and also learning opportunities available. Together, we can drive forward the innovations needed to combat cancer and also improve patient outcomes. So thank you for being here with us today, and I hope you all have an inspiring and productive day ahead. Thank you very much.
Thank you, Professor Ma. Please be seated. I would now like to invite Professor Alice Wong, Associate Vice President, Research, Interim Director, and Chair Professor at the School of Biological Sciences of the Faculty of Science of the University of Hong Kong, to deliver opening remarks. Professor Wong, please.
Thank you.
First of all, thank you, Stephanie, for inviting me to give the opening remarks. Good morning, everyone. Professor Junyu, Stephanie, Carmen, Kevin, Kate, colleagues, distinguished speakers, students, fellows of the Yan Academy of Sciences, ladies and gentlemen, welcome to Hong Kong U. And also, of course, welcome to the fourth sub-conference of the Yan Academy of Science Summit. And this year, the theme is From Cancer Discovery to Therapeutic Innovation. So as Stephanie mentioned, I'm also a cancer biologist. So as a scientist and cancer researcher, I find that we are engaged in very meaningful work here. Today, we are gathering here not only to share our latest findings, but to also inspire collaboration that will drive innovation and breakthroughs in cancer diagnosis, prevention, and treatment. So I'm excited to actually see speakers from different institutions here and from various disciplines, including biologists, chemists, clinicians, data scientists, engineering, and I also know that some of you are entrepreneurs here as well. So to tackle the challenges that cancer presents, we actually need interdisciplinary efforts. So we must harness the collaborative expertise, but also at the same time, fresh perspectives of the young scientific community to push the boundaries to what it is possible now. So from exploring novel therapeutic approaches to leveraging on some of the cutting edge technologies nowadays like AI and genomics. So we hope that we will make a real and significant difference. So as Stephanie said, I would also encourage you to engage with the speakers, ask questions, and take full advantages of the discussions. So let us use this summit as a platform to inspire each other, forge new partnership, and lay the groundwork for a future where cancer hopefully is no longer a life-threatening disease, but being a manageable conditions. So thank you all for being here today, and wish you a fruitful and inspiring conference today. Thank you.
Thank you, Professor Wong. Please remain on stage. We would like to invite Professor Wong and our guests for a group photo. May I invite the following guests to the stage, please? Professor Stephanie Ma, Professor Kevin De, Professor Carmen Wong, Professor Chen Kit Kwok, Professor Yu Jun, Professor Li Xiang David, Professor Wei Leung Tam, Professor Ng Lai Guan, Professor Terence G.Y. Wong, Professor Zheng Zhong Li, Professor Rebecca Teen, Professor Alan Siu Lun Wong, Professor Wu Zhenqing, Professor Clive Yik Shum Chung, Professor Zhou Jingying. To capture this wonderful moment, may I invite all our guests to stand closer to each other and face your left and right sides towards the camera. Yeah. So we can capture a wonderful moment and get a great angle of everyone. maybe let's put a thumbs up to record this memorable moment. Please keep looking at the official camera. Thank you very much. Please be seated. To record this event, may I invite everyone here to also take a group photo. Please remain in your seats. Our photographer will take pictures from the stage. So may I invite everyone to please look at the camera here. May I invite everyone to look at the camera with your big smile again. And let's give a thumbs up as well. 1, 2, 3. Thank you very much. Now, let's kick off the first session of the conference. This session, our scientists will discuss topics about prevention and early detection of cancer. Reminders to all attendees, during the session, please do not photograph or video record the presentation. Thank you for your cooperation. For online audience, due to the confidential contact, the live streaming will be temporarily suspended after the sharing session by Professor Terence Wong. We will be back after the lunch break. Please welcome the moderator of the first session to the stage first. Professor Chun Kit Kwok. He is a member of the Hong Kong Young Academy of Sciences, Associate Professor of the Department of Chemistry of the City University of Hong Kong. Professor Kwok, please.
Thank you, everyone. So let's kickstart the session. So we are on theme 1. We have 3 distinguished speakers today. Maybe I will first introduce Professor Yu Jun from, she's a member, well, I'll just read from my, so she's a member of the Hong Kong Academy of Science, jointly Professor of Medicine and Therapeutics, and she's from Chinese University of Hong Kong. So please join me to welcome Professor Yu.
Good morning, everyone. Thanks for the kind invitation from Professor Stephanie Ma and also the organizing committee. It's my great honor and pleasure to share with you a topic in terms of microbiome in gastrointestinal cancer, because the theme of this conference or summit is focused on diagnosis, prevention, and treatment. So I will focus on the correlation between microbiome in gastrointestinal cancer from basic research and molecular investigation and translate to potential diagnosis, prevention, and treatment to meet the main theme of this summit. From this picture, you can recognize that the whole human gene, the whole body, the host gene just account for 1%. About 99% are microbiome. So this is quite a huge amount of microbiome, how they play an important role in health and disease, attract more interesting and for scientific research in this particular area to link with the health and the disease, not limited to cancer. In terms of microbiome, in considering the location, we would like to say perhaps in other cancer type, the close correlation interplay is mainly belong to gastrointestinal cancer. Let's mainly study 2 cancer types belong to GI. One is a colorectal cancer, a second one is a gastric cancer. Whether they have a correlation or whether the microbiome is a cause or consequence, whether microbiome play an important role in the initiation progression of gastrointestinal cancer, actually is a certain issue. We need to evaluate and study step by step. Let's start from the mechanism investigation, the interplay between change microbiome and GI cancer. And then we move on to potential clinical application. before we work on the correlation between microbiome and cancer, we would like to have a screening. We have a different cohort from Hong Kong Chinese, from Germany, French, US, and also Austria. We want to identify common feature because microbiome, especially gut microbiome, easily affected by different parameters. For example, the diet, environment, the external issue, they all can change the gut microbiome. To identify the common feature, whether gut microbiome is correlated with colorectal cancer, we have to do multiple population to identify the common feature. And based on this result, you can see that. And this one is the CRC from Chinese Hong Kong, and this is the control. The composite microbiome are quite different, and based on shotgun mechanism sequencing from stool samples. In particular, we have identified commonly enriched bacteria in the red dot in multiple populations, Chinese, Germany, and France, US, and Austria. And we also identified depleted bacteria. Oh, it's not, cannot hear see there. Okay, and perhaps use this one. No laser? Okay, no point, it's fine. And then you can see the green, the green color is depleted bacteria, red dot is enriched. And this is based on the stool, and just the stool in CRCS compare control. You cannot give an answer as a cause or consequence. To answer this question, we have the multiple biopsy from different cohort as well. In control, you can see the paired adenoma is a precancerous lesion, is the benign. Okay, a benign mass polyps, and then paired CRC. So paired adenoma, paired CRC to see what's happened. You can see the diagram and the interplay among the microbiome from normal adenoma to CRC. There is a progressive shipment, a progressive change. This suggests that the change of microbiome is perhaps not a consequence, it's an early event. Then we would like to answer whether changing of microbiome do play some role in contributing to the colorectal cancer. And then to answer this question, we pull the stools from CRC patient, still remember the shotgun mechanism sequencing, the different abundance from colorectal cancer patient CRC as compared to healthy control. We pull the stools from colorectal cancer and then compare with the healthy subject gavage to different mouse model, especially the germ-free mouse model without any other treatment. The CRC stool can trigger the initiation progression of colorectal cancer, especially at a different time point. It can trigger the enhance the cell proliferation, enhance the expression of pro-inflammatory factors and also the oncogenic gene expression. And based on this observation, based on this result, we among the first to report that the colorectal cancer stool is toxic, it's oncogenic, can promote the initiation progression of colorectal cancer. This all composite microbiome and contribute to colorectal cancer initiation progression. And we do the different layers study. We identify some bacteria, fungi, archaea, virus. They have a different role in promoting initiation progression of colorectal cancer in separate study. And then we move back to the total microbiome. Is there any driver bacteria? Perhaps they put a very predominant role in contributing to the colorectal cancer initiation and progression. And then we focus on the enriched bacteria in the multiple population, the common enriched but not unique in individual population, the common enriched, perhaps they do play some role in colorectal cancer progression. And based on the enriched bacteria, only Fusobacterium nucleatum was reported by culture, this bacteria reported to be associated with colorectal cancer. Other bacteria, they enriched, but no one knows the correlation and between that bacteria in colorectal cancer. Then we choose one example. It's a Streptococcus anaerobus. This bacteria is a significantly enriched multiple population, and then we verify this bacteria in second cohort or third cohort in tissue biopsy and also additional stool samples in colorectal cancer, adenoma, and control to see whether this bacteria still enriched in different population indeed. Not only limited in stool, also in tissue biopsy, this by enriched in adenoma, further enhanced in colorectal cancer, and perhaps this bacteria contributing to the disease progression. And Frankie was a postdoc fellow, was working with me a few years ago, and then we chose this bacteria to further study. And at that time, no one knows the correlation of this bacteria with cancer. Only some publication shows that this bacteria can induce infection. And then in Frank's study, he demonstrated that bacteria gavage to mice and induce colorectal cancer or contributed to promote colorectal cancer initiation and progression. Steph is my second postdoc fellow. We do the magnetic investigation, how this bacteria can contribute to colorectal cancer. You can see under this electron microscope, in the colon cancer cell line, and more bacteria touch the epithelial cell, invaded the intracellular of the colon cancer cell line. But in the normal control, okay, epithelial cell line, it's just attached, not that frequent, it's very less. So it's the bacteria like the cancer situation. And then we evaluate the mechanism of how this bacteria interplay with the host epithelial cell. We're demonstrating that the surface protein from this bacteria, PCWBR2, binding to the receptor integrin-2 beta-1 from the host. With this binding, this bacteria can promote PI3K-AKT, you know, the oncogenic signaling pathway activation can promote the cell proliferation. And this gives an answer in terms of magnetic investigation, how this bacteria and then promote the cancer initiation progress in colon. And then the method we published in this paper, and the following the other studies, other groups, they follow this methodology to identify the surface protein from bacteria and the receptor from the host cell, and based on the different investigation and different diseases. So this is a kind of a platform or method and provide for the public different research group can follow to identify the surface and receptor from bacteria and from the host. At that time, we just did the single-cell sequencing, and based on the single-cell, we identified MDSC tumor-suppressive MDSC, a tumor-associated macrophage, enriched in different mouse model. But we did not dig up further how this bacteria can modulate immunotumor-suppressive microenvironment. So, but just observation for such kind of data result based on single-cell sequencing. And then this is my PhD student and postdoc fellow right now is working on this bacteria for 4 and 5 years. And then she further to study how this bacteria can, activate or increase the infiltration of MDSC in the tumor site in colorectal cancer. And then she demonstrating that there are 2 arms, not a single arm. One arm, we still remember, this bacteria can bind into the receptor integrin-2 beta-1, and then to activate the PI3K-AKT. And PI3K-AKT can further activate the RKB signaling pathway. With the activation of RKB, it can release the CXCR1, and CXCR1 release and then can bind into the receptor of MDSC CXCR2. With this binding, and then the MDSC can be recruited to the tumor site. And on other side, another arm, because it's just recruit MDSC in the tumor site, but cannot activate MDSC. And then we're looking for whether this bacteria have other connection with this MDSC, and based on the screening and different kind of searching or identification for the link and between this bacteria with MDSC, with them treating that this bacteria produced RYTC22, this enzyme, a protein. And this protein can bind into SLAMB4 on MDSC surface receptor, can activate MDSC. And then we evaluate this bacterial infection, P. anaerobus infection, in the mouse model. Because we use MSI high model, it should have response to the anti-PD-1. But if we gavage this bacteria, the response will be loose. It changes to resist to anti-PD-1 therapy. On the other side, we work on to block these 2 receptors, integrin-2 beta-1, and block SLAMB4. Then interestingly, and that resistance situation can be reversed to sensitivity in terms of anti-PD-1 therapy. And what Steph is still working on in terms of this bacteria, and especially this bacteria and in the interplay for Stamnase, and then we identify this bacteria also plays some role. If the pathway is different as compared with modulation of microenvironment, and this bacteria do have some function linked with Stamnase regulation in colorectal cancer. The work is and close to be finished, but still have some additional bacterial investigation, especially to knock out the critical gene and then play an important role linked with the Stamnase. Still this is a very, very critical part. We try almost a year, but still work on that. Hopefully we can knock out successfully and to link clearly in terms of this bacteria in correlate with the Staphylococcus. This is another example. It's also enriched bacterium colorectal cancer, but this bacteria also have a tight attachment to the epithelial cell, but it activate another kind of oncogenic signaling pathway and to promote the initiation progression of colorectal cancer. This is just an example for the bad bacteria in colorectal cancer. They do have a bad role, and because they are enriched, and then they promote colorectal cancer. And then we have another candidate bacteria. In additional colon cancer, we work on gastric cancer for another example. And for the gastric cancer, we want to see whether in gastric cancer, we do have the microbial alteration according to the disease progression. We have the biopsy from superficial gastritis, atrophy, and intestinal metaplasia, and to gastric cancer. 2 cohorts as well. One is from Xi'an, one is in Mongolia, because we want to identify common feature, but not unique feature. You know, that lifestyle and the diet are quite different from these 2 regions. Atrophy and intestinal metaplasia is precancerous lesion and before gastric cancer. Because it is a tissue biopsy, we did a microbial profiling by 6S sequencing. You can see according to the disease progression, superficial gastritis, atrophy, intestinal metaplasia, gastric cancer, the interplay and between among the bacteria, they change and gradually. And then this suggested that these bacteria may play some early role at the initial stage and precancerous lesion stage. We compared a different composition of bacteria in gastric cancer as compared with superficial gastritis. You can see, and a lot of the yellow color enriched in gastric cancer. That's enriched bacteria in colorectal cancer. And then some are enriched in superficial gastritis is a control. So based on the enriched bacteria, we identify Streptococcus aeruginosus. It's significantly enriched as the top bacteria enriched in gastric cancer. We put these bacteria here, and then on other side, we do another study. You know, H. pylori is the first carcinogen according to WHO. We want to see, in addition to H. pylori, if there's any other bacteria may contribute to no. Sorry. So. Okay, so we start here. Perhaps this one? Yeah, okay. And you can see, and this is from the disease progression. I move on to the gastric cancer, another example. So, and from superficial gastric atrophy, intestinal metaplasia in gastric cancer, we have 2 cohorts and confirm that the bacterial network, you can see the co-occurrence or co-inclusion or correlation is a change from the steps disease progression. So occur in precancerous lesion, atrophy, and intestinal metaplasia before gastric cancer. So, and this result suggests that the bacteria may also involve in the progression of gastrocarcinogenesis. And then we compare the enriched or depleted bacteria identified in gastric cancer mucosa as compared to superficial gastritis. They do have some enriched in the red window and also depleted in the blue window. So in terms of enriched bacteria, identified Streptococcus aeruginosus is significantly enriched as one of the top bacteria in gastric cancer. And they want to see whether after H. pylori eradication, if there's any other, because H. pylori identified in 1983, since H. pylori identification, no other report to identify other bacteria perhaps also contributing to the gastric carcinogenesis. So we want to see whether without H. pylori, there's other bacteria can also contributing to gastrocarcinogenesis. This in 2000, May, and led by Professor Joseph Song, we went to Shandong Muping. This is a high-risk area for gastric cancer. We do the population-based study to eradicate H. pylori and the placebo. We want to see if eradication of H. pylori, we can identify the disease progression because we follow up 1 year and 5 years. Without H. pylori, I want to see if the precancerous lesion or cancer can be suppressed significant as compared to control. And this doctor, yeah, it's me, about so many years ago, and perhaps you cannot recognize. in Shen, we have a team and from Prince of Wales Hospital, and yeah, to do the endoscopy very early morning, 6 o'clock we get up, start, because the farmer need to go do the farm work in the morning, so we finished in the whole morning. And these are two populations, the randomized study. You can see we do the H. pylori eradication, triple therapy, and the placebo randomly. If we detect H. pylori positive and randomly have two groups, treat or not treat. And then we follow up a year. I just show 1 year result. We do have some samples for 5-year follow-up. We want to see what's happened without H. pylori, and then do the screening, microbial screening, and the pathologist do the evaluation in terms of histology. Without H. pylori, the disease still progressive. So we identify, you can see the red column. This is the bacteria correlated with the disease progression after year H. pylori eradication. And then for atrophy, and the lower column is the intestinal metaplasia. When we overlap with the enriched bacteria, you can see 4 bacteria, they contribute to the disease progression, and also significantly enriched in gastrocancer tissue. Still, Streptococcus aeruginosus occur again. It's correlated disease progression, and also enriched of gastrocancer. What's the Streptococcus aeruginosus? We did some literature review, and this bacteria can cause infection, the oral, and also the respiratory system, and also vaginal infection, but no report in terms of this bacteria correlated with cancer, including gastrocancer. So we choose this bacteria for study, and then after 2 weeks infection, it's induce acute gastritis, H. pylori as a positive control without other treatment. And all the histology evaluation and done by the chairman of the Department of Pathology, Professor Dou, and also the assistant professor Alvin. So not by us. Our team, all the histology evaluation, all passed to the pathologist to give us a diagnosis, liver, colon, and stomach. They did a great job. So after long-term infection, 3 months, 6 months, 9 months, 12 months, without any other treatment, germ-free or germ-conventional mouse model, and only this bacterial infection can induce chronic gastritis, atrophy, metaplasia, and even dysplasia. So if with the, the carcinogen, MNU-induced carcinogenesis with this bacterial infection can promote the tumor number or tumor size and gastrocancer formation. And then we do the mechanism investigation, how this bacteria play a role to initiate the and promote gastrocarcinogenesis. And then we identify the surface protein, bacteria, TMPC, and binding to the receptor anatium A2 at acute situation induce the pro-inflammatory release, induction of the CCL20, CCL8. But in long-term infection, after 3 months, 9 months, or 12 months, and this bacteria attached to the epithelial cell can increase the oncogenic pathway, MAPK, GNK. And also, if we knock out the TMPC, and then the function of the bacteria initiates chronic inflammation, and also gastrocarcinogenesis can be totally abolished. So this, in addition to H. pylori, another bacteria, we identified that correlated with the gastric carcinogenesis. The work is still going on. We have different angles. And then we're working on other effects of this bacteria contributing to the stomach cancer. So this is basic research in terms of interplay, magnetic link, and between the selective representative bacteria with colorectal cancer and gastric cancer. Let's move on to the main theme for this, our summit for today, and prevention, biomarker, and treatment. So we discuss with the prevention first. If we talk about the prevention, we'll link to the left-hand side, the depleted bacteria in common in multiple population. So for example, Streptococcus thermophilus is a depleted bacteria in colorectal cancer as compared with healthy subjects. What that? And then the triangle is the known probiotics. So these known probiotics, no report for the link with the cancer prevention. You see the happy face and bacterial Gavaggio different mouse model, it can suppress colon cancer formation. So we work out the molecular link, how this bacteria can suppress the colorectal cancer is mainly because this bacteria can produce beta-galactosidase. This enzyme, it can produce more galactose, and galactose can suppress Warburg effect and therefore suppress colorectal cancer. And then if you knock out the LAC gene, cannot produce beta-galactosidase, and the bacteria, the protective function will be totally lost. That means bacteria produce beta-galactosidase is a major or maybe the only functional product in suppressed colorectal cancer. And this bacteria is good, no gender difference, and then for male and female, they all play a good role. And then we identify bacteria, say Mathematicum, and this bacteria depleted in female CRC, but not depleted in male CRC. Why is it depleted male CRC? We do the mouse model. It's also depleted bacteria. And this bacteria only function in female mice, but not function in male mice. And then we'll work out if we remove the ovarian from the male mice, the female mice, and the female mice, the protective function will totally lose. So that means, and this bacteria, and then have a female, perhaps an estrogen-dependent fashion, we then link with the bacteria, whether they have any potential interplay with estrogen. We demonstrating that because the bacteria need to colonize in the epithelial cell in colon, the receptor SLC3A2 is the estrogen-dependent fashion. After bacteria colonize in the colon epithelial cell and based on the receptor SLC3A2, the bacteria can produce some product metabolize 7-DHC. It can convert to vitamin D and then to suppress the vitamin D signaling pathway, therefore suppress colorectal cancer. These are two good bacteria depleting colorectal cancer. We have a package of depleted bacteria in colorectal cancer. We apply the patent and license it to a local company. It's a package for beneficial bacteria. It's a disease-specific or colon cancer-specific good bacteria, and have a package. The local company is doing the clinical trial. The year 1 clinical trial has been finished because in colorectal cancer patients before colorectal cancer, long term or many years for polyps, adenoma, and this is a benign neoplasia, but it takes maybe 10 years, 5 years, 18, no one knows. But always based on the screening, colonoscopy can remove all this neoplasia for polyps, adenoma. 3 years or 5 years, the recovery is up to 60%. It come again, always. And then the doctor will tell you, and come to me to the colonoscopy screening. And then because last weekend, I went to the 6th hospital, Sun-Hsiang University. The dean of the hospital is an expert, especially on colon and intestinal disease, as a surgeon. He also mentioned that no way to do himself as well, and after colonoscopy screening and removal. After a few years, he follow up more frequently, and 3 years, a new one come out, and then removal again. No other way. So we hope if these probiotics do have some function to suppress recurrence, and this clinical trial is doing to focus on the removal of polyps adenoma, because very common, and then to see whether it can suppress relapse for the polyps and adenoma, and based on the supplement of these probiotics, if they do a good job and long-term can suppress colorectal cancer formation. So this is the purpose from microbiome research for the clinical translation for cancer prevention. So here is the cancer prevention. Let's move on to the biomarker. So we see that the differential abundance of bacteria, and based on, still remember this picture, is some bacteria enriched in colorectal cancer, some bacteria enriched in healthy subjects. We demonstrating that the 20 bacteria genome can distinguish colorectal cancer versus healthy subjects. And this biomarker, 20 gene biomarker, independent type 2 diabetes, inflammatory bowel disease. Perhaps you can ask why you need to distinguish type 2 diabetes, inflammatory bowel disease, and because of the high risk in contributing to colorectal cancer. We also did gastric cancer, and this marker, independent gastric cancer as well. So, and then we study these bacteria to see whether they have a good role for the diagnosis early colorectal cancer, because later stage, when they see a patient with a symptom, is always at the advanced stage. So we need to identify screening at the early stage. This is a PCR-based detection. It's low cost, and also high sensitivity, high specificity. So compared with the FIT, this is a clinical commonly applied screening for colorectal cancer. The FIT is a green color. The blue color is the bacterial marker. For stage 1 and 2 and 3, the bacterial marker in terms of sensitivity specificity is compared with FIT overall have a good performance. And we also identify a bacteria named M3 because it is a new bacteria. So we gave a name M3 is good for the advanced adenoma detection. We also package this bacteria for marker, and then we apply the lesson and transfer to a local company. I focus on research. I do not have any function or role in the company because I think I would like to focus and more focus on research. So I just licensed to different company. And so this company, they established the M3 detection kit. Now it's applied in Hong Kong and then Asia Pacific region in private hospital or public hospital, I think in Hong Kong as well, and do this early detection or screening for colorectal cancer and advanced adenoma. And this is another translational example in terms of microbiome research for cancer detection. They also apply NMPA in mainland China. Perhaps they get approval. They are still working on that approval, not too long. And for this bacteria-based biomarker for the screening, advanced adenoma, and colorectal cancer. This is a bacterial research. We want to see if the metabolomic also plays some role, can be a marker for the cancer detection. And this is a recent publication. Jennifer is here, and Jennifer is the co-first author by work with, because the first authorship, they help us to collect a lot of samples from Kunming. We have the 4 cohort in terms of, a discovery cohort and 3 validation cohort to prove, to confirm this metabolomic biomarker in colon cancer have a good function, a good role, and then for the detection. We did the shotgun measurement sequencing for stool. You see the large samples in total is 1,281 or 51. Cannot see, it's too tiny. And the samples for the targeted first started non-targeted metabolomic assay from plasma and also stool, and then move on to targeted metabolomic assay. So we're very expensive for the screening, and then in terms of biomarker and also the functional validation. You can see, compared with the stool, the consumed metabolites, the plasma metabolites is more pure, more clear as compared with stool, more stable. So this is the plasma metabolomics. You can see from normal control, CRA is adenoma, CRC is colorectal cancer. They are increasing trend for the enriched metabolites and also declining trend for the reduced metabolites according to the disease progression. And based on this heat map, in terms of metabolomic difference, we had the middle stage. We identify some biomarker for this metabolomic biomarker based detection for colorectal cancer. You can see the colorectal cancer as compared with normal control. The AUC can over 0.9. And also the early CRC stage 1, 2 versus control, the AUC is still 0.958. And the CRC and compare the adenoma, they also have a good AUC, and especially CRA, the early stage, the adenoma compared to healthy control also show a good AUC. Compared with FIT, the commonly fecal blood detection, and this CRA versus control have a better performance. And this is a metabolomic marker. The company, and because the mainland company applied a patent and do the validation in different cohort and before applied, got the approval from NMPA in mainland China. So this marker is still going on to form a detection kit and based on metabolomic alteration. This metabolomic alteration is only focused on bacteria-associated metabolites, but not the host, not the food. We exclude the food metabolites and the host metabolites and focus on microbial metabolites. So to summarize our marker, we are not only working on the bacterial marker, metabolomic marker, we have a history to work on the marker. So we did the publication microRNA detection kit. It's the first stool-based molecular detection approved by China FDA or NMPA, as approved in 2018, now applied in the mainland China, different hospital for the early detection of colorectal cancer, and based on the PCR detection for microRNA 92A. And then the second marker got approval for China FDA and MPC, the new name. And this is a detection marker, a methylation marker RF180 for gastric cancer. This is also a first MPA approval biomarker, molecular biomarker for gastric cancer detection. This got approval in 2020. And M3, I do not like to repeat, and this Hong Kong marker approved in the clinical practice. And so these 3 markers all got approval, now applied in clinical practice in mainland China, some in Hong Kong, and Asia Pacific region. Last, finally, I don't know if I have some time or not. Sorry. Can I continue or just stop? So I quickly finish. So let's see the therapy and the immunotherapy. And this is a very hot research area, and a lot of people is working on this area. We want to see the change of gut microbiome metabolites can change immune cold tumor to hot tumor. So we want to have this question. And this idea is mainly from in 1999, a 2 publications from melanoma. They do the fecal microRNA transplantation from the stool from responders to non-responders. Then certain number of patients can from non-responders to responders. So microbiome metabolism may play some role. And these are 2 publications in colorectal cancer, and also perhaps the first publication, we identified 2 bacteria can modulate MSS, microcell stability stable, colorectal cancer, and because it's this non-responses to anti-PD-1 therapy, and then after the gavage this bacteria and the bacteria metabolites one bacteria, the Superior intestinalis can directly increase and activate the CD8 T cell by producing butyrate. A second bacteria, Alloglinarum, and these are good bacteria by suppress Treg indirectly to activate the CD8 T cell to suppress colorectal cancer. And this is a very recent study, and this study and fusobacterial nuclear atom is a bad bacteria. We use the humanized mouse model, different germ-free or conventional immunohumanized mouse model, and confirm that this bacteria in modulating immunotherapy can induce anti-PD-1 efficacy. So I may not give detail, mainly suppress the CD8 T cell, the surface PD-1 expression, and through histone modification. And with the suppression CD8 positive surface and PD-1, they can change how the CD8 T cell to active the CD8 T cell. That's why can increase efficacy of anti-PD-1 therapy. Just to summarize the study, I do understand the study for microbiome research from screening, academic investigation in recent years, a couple of years. You can see the invited review article. They more highlight the prevention and treatment. So still have quite a lot uncertain question. We need to still spend more time to dig up, especially for chemotherapy, radiotherapy, adjuvant therapy in addition to the immunotherapy, other therapy is also quite important. This group photo, just 2 months ago, we have the annual report for Sticky Lab and also the Institute of Digestive Disease. So thanks for all the clinicians, mainly in first row of scientists and researchers in the back row for their great contribution to the individual study from Institute of Digestive Disease and from Sticky Laboratory of Digestive Disease. And before closing my session, I have a poster to most welcome for the young faculty members and to come to join our International Digestive Disease Forum. And this year will be in Conventional Center 5 to 6 July. And last year we have over 1,600 delegates from over 30 countries. Hopefully you can join us and submit your abstract. The end date is March 28th. Thank you so much for your attention. Thank you.
Thank you, Professor Yu, for a wonderful talk. So we'll have some Q&A session after the whole section. So please ready for your question. So now we should move on to the next speaker. So the next speaker is Terence Wong. He's an associate professor from the Department of Chemical and Biological Engineering from the Hong Kong University of Science and Technology. Please join me to welcome Professor Wong.
I guess I know how to do it.
Yeah, thank you. Okay.
Should we start the clock? Oh, it's not. All right, so good morning, everyone. Thank you very much for the very kind invitation from Stephanie, Kevin, and organizing committee. So this is Terence from HKUST. So I'm very glad to share with you our exciting work, including 2 integrated approaches on medical diagnosis on cancer by integrating, first of all, AI, and also next generation histological imaging. And before I start, I have to declare that I do have a startup company trying to translate or translational research from what we have in the lab to the hopefully in the hospitals in a very short period of time. So my lab is focusing on the co-development of 2 things, mainly corely. So imaging science, we do have imaging technologies like photoacoustic imaging with or without labels, optical imaging approaches, also with or without labels, and also with the integration of artificial intelligence. Nowadays, generally saying there's AI, but specifically we mostly focus on deep learning. So by integrating these 3 things, our key, very core mission is to improve healthcare system with better technologies. So let's go through a journey. What exactly the problem we are trying to solve? Let's say this is a cancer patient. He or she may not know at the first beginning, maybe just feeling kind of sick. And then the first way first approach of solving this problem is to maybe seeing a medical doctor, maybe having a body checkup, getting some indexes from your body, and then trying to do more investigations. What are the more investigation could be radiology. So macroscopically knowing the conditions about your body, like X-rays, MRIs, and also ultrasounds. So all those are just trying to get a rough sense about your health status without opening up your body. And there's another approaches, I think we also encounter that very much in the biomedical sciences, is that histopathology. And histopathology is the very accurate diagnosis we can make based on these pink and purplish color images called H&E stain images. So in particular, in this talk, I will be mainly focusing on the bottom row, the histopathology. I think I have like 1/4 of my lab doing the top row, the radiology part, but the main cohort of my lab mission is drawing from the bottom row. So what exactly is histopathology? I guess not all of us are expert. Maybe there will be some teenagers, young scientists in doing this field. So I have very brief introduction about this histological imaging. So this kind of histological images, you basically will have that when you examine your tissues under microscopes. So what are the functions of it? It's very, very, very obvious to many of us, is to use to classify many different type of diseases like cancers or inflammations or just normal, so you're good to go. So you can also check the stage and presence of cancers, and eventually what's the most important thing is to guide the doctors to provide the best treatment for the patients. So nowadays, what are the options in the hospitals? Mainly 2 routes. The first one is during operations, intraoperative use, you use a method called frozen sections. Frozen section is very fast. It just takes around 30 minutes, you get a results. So the bad thing about that is that the image quality is in generally low and inconsistent, not to mention that some of the tissues just intrinsically hard to deal with, like fat and also water-rich tissue. And another approach is post-operative use when time is not so important. You can wait a little bit more. Then this FFPE formalin fixed paraffin bedding is really the gold standard. You get the best image quality, general all the tissues applicable, but the key issues is that it would take up to a week. So this could be a problem, right? So because you see that one approach is kind of fast, but relatively inaccurate, not able to handle all the tissues. Another approach is kind of slow, but very accurate, so-called gold standards. So let's go through a case study, what exactly the problem we are facing in the hospitals. We call it cancer margin analysis. So just imagine I have breast cancers, okay? So a patient, male can also have breast cancer. So let's imagine I have breast cancers. Many of the patients in the US in particular, they will go for conserving surgeries. What does it mean? It means that we only cut out the problematic cancer cells and try our very best to preserve the normal cells as much as possible. So we can see the bottom cartoon here. The blue dashed line is the excised tissues. And then you see the case on the left is so-called a successful cut because all the cancer cells are being encapsulated and removed from your body. But then for the positive margins, is really after the surgery, there are still cancer cells remain after the surgical cut. And what's even worse is, it's not a little number. 20% of the patients undergoing conserving surgeries end up with the case on the right. So 1 out of 5 patients end up with the case on the right. And they only know it after a week of doing their surgeries. So we definitely need a better diagnostic tool to address this issue. But how come with the current two technologies not able to solve this problem? Let's go through the very detailed investigations. So let's say this patient undergoing surgeries. Nowadays, they use frozen section to confirm whether they excise all the tissue, I mean problematic cancer cells out. And then in about 20 minutes time, the pathologist will be able to tell the surgeons whether he or she did a good cut in the initial surgeries. And it could be a loop. I mean, maybe this first tissue is not clean cut, send another one. Second tissue not clean cut, another one, until all the cancers are successfully removed and being diagnosed and checked with frozen sections. And the same tissues, remaining tissues, will go through the 1-week test, the very accurate FFP test. And the dynamic is really here is that if we want to have a very accurate test result, they may be able to check there are some residual cancers in your body. So the tragedy is that it will call for a second surgery if the FFP test result is actually positive. So, but then for sure the patient is already good to go when the frozen section result is negative. So the problem arises from the fast but inaccurate test result and with the combination with the slow but accurate test. So we frame it as a research questions now. So can we keep things simple while getting high quality images like FFPE, that means the very accurate test result, while it's so fast? Okay, so that's the goal. So we envision in this way in 2017, and then we try to harvest the tissue and directly image the tissue without any processing. We generate image that is so high quality that pathologists can understand. So that's also after that year, I mean 2018, I went back to Hong Kong, joined UST, and start investigating how can we solve this problem. So this is the entire journey that the sample will go through in order to get the very accurate test results. And then it classified into some sample processing steps, staining step, and manual diagnosis steps. So we are trying to see how can we speed up each of these processes. For example, can we do slice-free imaging? We don't want to slice the tissues. We just want to image the tissues that is freshly cut from patient body. And we also see if we can use AI to do the actual staining, I mean the virtual staining with replacing the actual stainings. For sure, we can combine both, right? So we just directly harvest the tissues, image it, and directly generate images the pathologist can understand. And nowadays, not surprising, we also can use AI diagnosis to further assist the pathologist to make a more accurate diagnosis and more effective diagnosis. At the end of the day, what we are preparing also is to have starting from the sample tissues, we directly go to the end goal. So from the image, from the tissues, we put into our machines and generate results that already telling the surgeons whether he or she did a good cut. So for the interest of time, I cannot go through all the publications, obviously. I just highlight 4 of the works and share with you. So what is the key problem of slice-free imaging? I guess many biologists and many medical professionals here, most of the microscopes is very afraid of thick tissues. I mean, tissues that's not optically thin. It's because dealing with thick tissues, they are intrinsically having many layers, information stacking up together. So we're intrinsically trying to solve a problem that hopefully we have imaging methodologies that can image thick tissues without physical sectionings. So I guess many of you know laser scanning confocal microscopy. We just try to play with light properties to select one layer, and that's good to go. And then we also have photoacoustic microscopy, and this is also one of the work that brought me back to Hong Kong. And we also have nonlinear microscopies. All these are really playing tricks in optics and somehow to select a particular layers, very powerful. However, all these three methods have a very important drawback, is that they are label-free, don't need to perturb the tissues, don't need to destroy the tissues also. But then the imaging speed is fairly low because we do point-by-point scanning. I guess many of you use laser scanning confocal. When you press the scan button, you better go for lunch, right? So you don't want to wait for such a long image during surgeries. So we then go for another approach and look at this problem. Can we do wide field? So just turn on the lamp and then we get a 2D image. That's the dream. So light sheet microscopy, like with fluorescence, is one of the options. Structural illumination microscopy is one of the options. And also one methodology called microscopy with ultraviolet surface excitations, MUSE, is also another option. So all these three methods in general are in plane. I mean, get an image every time with a 2D image. But the problem is that all these three methods requires fluorescence labels. If you still remember what I envisioned about the future is that we try to cut out the tissues and image directly without any perturbation on the tissues. So we are proposing to integrate two sides. I mean, we do have the wide field imaging. At the same time, we use some other tricks to bypassing the fluorescence staining. So we propose a methodology called computational high throughput autofluorescence microscopy by pattern illumination, CHAMP in short. So this method does not require tissue sectioning, does not require tissue stainings, because we intrinsically play with some optical properties with biomolecules, such that they have fairly good fluorescence and fairly dark fluorescence. That means they do not fluoresce and provide intrinsic contrast between cell nuclei and also the cytoplasm, mimicking H&E stainings. And we also play with optical tricks, and then we enjoy large imaging field of view and resolution together. Normally, this is a heavy trade-off in optics. And last but certainly not least is that we have a high tolerance to tissue surface roughness with a long depth of field. This is also very important clinically because we're dealing with a tissue that is freshly cut from surgeons. You just don't have time or chance to ask him, hey, sorry, it's not flat enough, right? So can you cut a flatter slide? You just don't have a chance. So you have to have an engineering approach from the imaging perspective methods that is more tolerable to tissue surface roughness. And I believe you may not be able to tell what exactly this image is, but you'll be able to tell that the left one is more in focus entirely compared with the conventional method that is imaged with higher NA lenses. So that's not the end of the day, just the beginning of the story, is that because we generate images that is not commonly used by pathologists, grayscale. So grayscale has a lot of information, but just pathologists are so well trained to interpret H&E stains. So we don't want to have a paradigm shift on interpretation. That would cause a lot of challenges down the road if we want to do translational research. That's why we ask our computers to do a learning. So we call virtual staining a deep learning algorithm. We just input this left row and this right row images for the computer to learn what are the differences between them, maybe morphologically or maybe colorized, what kind of features they can see that are differences. And then after learning, we can input the left row, left column here, and then we can generate this middle column here. So in short, the computer learns how to do digital staining instead of real stainings. So the key here is that is just to make pathologists' life so easy. So a plug-and-play device in general and improve the clinical workflow without introducing a new standards. So then we use a virtual staining algorithm that trying to I mean, many researchers have tried. It's called unsupervised approaches. In particular, one method called CycleGAN, a GAN-based approach. It generally transfers a grayscale images to a pink and purplish H&E stain color images because we know this is the target, learning target. And the good thing about it is that these two images, you see, they are not even the same. I mean, so that's why we call it unsupervised learning. And it is commonly used in our approach because distortion is being normally generated in the sample preparation steps. Not to even mention that if you recall, our dream is to image a tissue that is freshly cut. And then when we image it, when you go through the histopathology process, when it rotates and you section it, it may even changing the orientations. So you just fundamentally cannot use supervised learning. So here we really don't need precise image registration with this unsupervised learning, so it's very good for our applications. And let me show you some data for lung cancers. So here's a thin slice images. Again, the left column here is the CHAMP, middle column is the deep CHAMP virtual stained, and the right column is the FFPHE. So I guess with the zoom-ins, we will be further convinced that the deep CHAMP in general generate images that is very similar to ground truth H&E, but this is just a thin slice. What we want to try is really a freshly cut tissues. So here you go for the results. So the middle column, again, is the generated images, but be very careful that they do not look exactly alike or the same. It's just like what I said, is because when I cut it out or go through the sample preparation processes, they will change in terms of orientations and also slice depth. So they won't be exactly the same. What we can do in the validation is ask a pathologist, give them these two sets of images, whether they can make the same diagnosis based on the tissues. So far we are running pretty well. So we have been working with quite a number of pathologists. The endorsement is pretty supportive. So seems that it's so powerful, and also the story is so good. And then when we try more, it doesn't work. Okay, so this is really science, right? So let's have some frustrations. We use some the unsupervised learning that we just proposed. And then we even try the state-of-the-art supervised learnings many groups have tried to use. But you see the ground truth H&E on the rightmost, they just do not echo with each other. So we try to investigate what exactly the problem is because I guess many of you have heard about deep learning, right? So the key thing about deep learning is that they need to learn enough different features, okay? And sometimes supervision is good, but just the pixel-to-pixel supervision is just too good to be true in our case. So we revisit this problem, and then we try to see if we can build something in between. So supervised learning, just said, is not good because the tissue intrinsically distort during the sample preparation process. And then unsupervised learning just did not take a full advantage of the fact that the H&E and also our CHAMP image do have some similarities, although may not be pixel-wise similarities, but then area-wise or region-wise, they could be still fairly similar. So we propose a new approach called UFrame, Unified Framework. We build a framework that is with a tunable parameters called a tolerance size here, the tolerance size, that will be able to tune the supervisions of these two images. I mean, how similar the two sets of images. So this is less requirement on data. Patch level alignment is already good, not pixel level. More importantly, we're also trying to ask the algorithm to sample some rare regions, because deep learning also need to learn enough rare regions, otherwise you have biased data. So here you go about showing what we have. Let me recall what we had just now is not a good ones with the supervised and unsupervised approaches. And with UFrame, you can almost going back to the ground truth H&E stains. I guess with the zoom in, you'll be convinced by yourselves without I trying to explain what exactly you're trying to see. In general, the cell features, it just pops up again. I mean, the lymphocytes surrounding the cancer cells, we will be able to tell them with two groups of cells in the images, and not only qualitatively eyeballing, but also quantitatively with a structure similarity index, and also with peak signal to noise ratios. So we then go one step further, is about medical diagnosis. So what we have just now in the last like 15 minutes, about past 15 minutes talk, is that we have the CHAMP image, we have the virtually stained images. nowadays in hospitals or in pathology lab, they do have a lot of these. I mean, they scan the tissues with H&E stains, and then they annotate the tissues where are the problematic cancer areas, and then do this kind of annotations process. And we can also ask computer to do that. But to do that, instead of doing these manual annotations, we can also ask just one question and push yes, no. Is that we input this virtually stained image and ask whether these slides contain any cancer or not. So yes, even if it's small cluster, you say yes. But if it's purely no, I mean completely clean, you say no. After training with more than 100 samples, you get something like this. I mean, it's a heat map showing high probability of the area with cancers in red and very low probability of having cancer cells in blue. So at the end of the day, what we need to do is just to compare the computer-generated heat map with the ground truth H&E stained annotated data with the pathologist input. So here shows you how we crack down the problem is that if we only have the AI diagnosis, sometimes it's just so hard and convince the pathologists and also medical doctors to use it. But when we have the heat map, the diagnosis results, together with the virtually stained images, they can still see the images. Hopefully we build the trust about AI and bring this on the table in a short time. So you can see the red area is the cancer area, and the blue area is just the normal area, which can be easily visualized with both the virtually stained HE and also the real HE. What I just said is only human. What do I mean by that? Is that we visualize colors like what we were trained. However, computers don't read colors like that way. So we also ask, is it possible to do general CHAM or autofluorescence image medical diagnosis directly? So this is what we have tried. And the surprising result is that we have only tried 120, around 120 samples, and we split them to training, validation, and testing set with 60%, 20%, and 20% splitting. And you can see that what we have found is that the AF, the CHAM image, actually have even better performance on predicting the cancer regions. That means echoing with what the ground truth H&E. So this is actually showing that the AF encodes even more information than the ground truth HE because we intrinsically see more biomolecules from the AF images. So here, also another way of looking at this is that without generating the virtual HE, we can also have the AF and have the heat map on top and compare with the ground truth annotated data. So with all these promises, we do have, previously we have more than like 200 samples collected with Queen Mary hospitals, Prince of Wales hospitals, and then we are expanding more. And this year, I think starting from next month, we will be sending our prototype, 5 of them, to different hospitals and doing this clinical trial in parallel. We are collaborating, as I said, Queen Mary, Prince of Wales, and recently we have already started working with HKSH medical group. And then we also have 3 more collaborators in China trying to boost the numbers. The goal is very obvious, try to boost the numbers of samples in a short time so that hopefully can validate and get the NMPA certification in a short time. So finally, what we are envisioning in the future is that during resection surgeries, we no longer need to rely on frozen sections. We can use these CHAM machines, load the samples into the CHAM microscopes, and then after a few minutes, I already disclosed how long it takes. After a few minutes, you will be able to generate an image, H&E image, together with the heat map, and remotely send to the pathologist who may be working at home or wherever. And then the pathologist can tell the surgeons he or she did a good cut, whether he or she did a good cut, in just 3 minutes. Okay, so because we don't need to protect the samples, we don't need to slice the sample, we don't need to stain the samples. So that's why this time is so fast. And then right now we are also speeding up the AI part, the algorithm part. Together, we believe that nowadays, what I know, the latest number is about 4 minutes, but we are shortening time to 3 minutes. And as I shared with you just now, the AI automated diagnosis already reaching higher than 90%. So we believe this will come true very soon. So this is what we have started for the resection surgeries. So very promising, but at the same time, whenever I talk to surgeons, he or she will tell me, hey, Terence, this is a wrong focus. He or she will say that you should focus on tissue biopsies. Because tissue biopsies immediately expand the scope of your project, and then you can have a lot of more reach of the tissues, and you can also do prevention of cancers because you advertise during early biopsies. Then you can also spot them, check them right away before they go back home. That will also bring a lot of values on the table. So with all the hard work, so we also have the startup trying to commercialize the technologies. So I have to disclose that. I already have that, and the company is called Formatics Limited. And recently, we also got supported in the Race Plus program. So I know that I only have 1 minute left, but I still have 5 more slides to go. So I will just, in short, summarizing what I have for the futures. So 2D H&E is just I share with you. But there are some groups saying that, wow, would it be possible to do even 3D? So as a scientist, we will say yes, right? For clinicians, they will say no. So as a scientist, I cannot say no, right? So we also tried it out for 3D histopathology. So for the interest of time, again, I will just tell you about the one work now because of time. So directly tell you the problem. So in the histopathology nowadays, they slice the tissue, they stain it, and then they get an image. And then if they want to stack a 3D, they are working on fragmented images. No matter how advanced the 3D registration algorithm, it doesn't work because sometimes the tissue is just ruptured when you prepare the thin slices. So we want to change, or we want to see if we can solve this problem by generating any distortion-free 3D histological imaging approaches. And that's why we came up with an approach called TRUST, a translational rapid ultraviolet surface tomography. So we are intrinsically, essentially flipping the sequence. So we have the intact tissues, and then we image it first, and then we slice it. So by reversing this process, so you will have the intact tissues always with you. You always slice the tissues that can be ruptured, whatever you name it. I mean, you don't mind. So, but then the remaining tissue is always intact, so you just stack them at the end. You don't need to have any registration algorithm, and you're good to go. So here shows you some high-quality images that we just simply stack them, and it's so generalizable. It can be workable for any types of tissues that we found so far. So for the interest of time, I'll skip this, but I will stop at playing this video for you that after generating all these high-quality images, we also do virtual staining, and then you can see this. We can have different cuts, different zoom-ins because of the high resolution, and yes. And we also have the negative contrast you can play with, so we have visualized the blood vessels also afterwards. You have a lot of room to play with. With that, I would like to end here and also give all the credits to my lab members because I'm always only the one speaking, doing that thing. They are always the one who do all the hard work. Thank you.
So thank you, Professor Wong, again, for the inspiring talk. So we have the last speaker for this session. So we have Professor Zhang Zhongli. So he is an associate professor in the Department of Biomedical Science at the City University of Hong Kong. So please join me to welcome